He will not force
Hello, everybody. Thank you for inviting me. It's an honor to give the Lord Florey lecture here at Lincoln College, Oxford. As a fellow Lincolnite, I'm honored to be giving a speech in honor of Lord Florey. For those of you who don't know who Lord Florey was, I will explain his career in just a moment. So today I'm going to be talking to you all about geroscience, which is the discipline of biology concerned with the fundamental mechanisms of aging and the role in disease processes. But before I get into that, I'll tell you about Howard Florey, the antibiotic revolution that he helped bring about, and its parallels to geroscience. So who was Howard Florey? Well, he was a pathology professor here at Lincoln College, Oxford, in the Dunn School of Pathology, and he ultimately won a Nobel Prize in medicine. To quote the Australian Prime Minister, Robert Menzies, he said, "In terms of global well-being, Florey was the most important man ever, asterisk, to be born in Australia." Australia having a population of 25 million, it's mostly empty space, but we'll let him have it. And here he is on the $50 bill in Australia, so he must have been important. But what exactly did Florey do? So in 1928, penicillin was discovered initially by accident by Alexander Fleming in London at Queen Mary. But it remained on the shelf, languishing and forgotten for over a decade. Then it was rediscovered in 1939 by Lincoln College fellow Florey and his team here at Oxford. In 1941, they conducted the first successful clinical trials on penicillin, just up here at the JR Hospital. In 1942 to 45, they scaled up mass production just in time for World War II. In what must have been the fastest turnaround ever for a Nobel Prize, in 1945, they were awarded the Nobel, shared between Florey and his colleague here at Oxford, the Jewish-German refugee, Sir Ernst Chain, and Fleming himself. So Florey helped usher in what is referred to as the Antibiotic Revolution from 1930, lasting until around 1980, when the pharmaceutical industry lost interest in developing new antibiotics. Although hopefully that will change as we have antibiotic-resistant bacteria cropping up even here in the Western world. So let's go back to the turn of the century. Around the year 1900, the leading causes of death were pneumonia, TB, and bacterial enteritis. That comprises 42% of all deaths being due to bacterial infection. Fast forward to today, the leading causes of death in 2018 are heart disease, cancer, stroke, lung disease, etc. These are non-infectious diseases. They're non-communicable. So what this means is only 6% of all deaths today are due to infection, and most of that 6% are actually viral infection. So what that means is less than 1% of deaths today are due to bacterial infection. That's at least for now, until bacteria continue to evolve antibiotic resistance. So hundreds of millions of lives were saved by Florey and his colleagues, maybe even your own, possibly my own life. So we have a lot to thank them for. So the argument that I'll make to you today is that the partial reversal of aging in humans, the ultimate objective of geroscience, will actually first occur in the 2020s sometime, as the first approved drugs for age-related diseases, but ultimately aging itself, will become available. And this revolution will be proportionate in magnitude or significance to the antibiotic revolution in the 30s. To better understand what the future of longevity will bring, you can read these highly acclaimed books by Yuval Noah Harari, Homo Deus and Sapiens, which both describe at some length the biology of aging and advances that we have made in recent years. So moving into the first part of my talk, I'm going to tell you about aging demographics and why it's so important now, today, to do something about aging and to attempt to slow the process. So we are facing what demographers call a silver tsunami, meaning we have an aging global population. And we're not replacing these people. So we're actually not producing enough to the replacement rate, so population is actually going to peak around 2050 according to the UN. So if you look at this map here, in the year 2010, the countries shaded in the color red are those where over 20% of the population is over age 65, or roughly the retirement age. If you fast forward to 2050, these UN projections show that pretty much all of the developed world, except for parts of Africa and India, will be dramatically aged. And the current medical system and pension system is not equipped to deal with this demographic crisis. So what's driving it is actually the baby boomers. The baby boomers are the generation that after World War II, people came home and celebrated and had kids. And they're now all starting to retire. That results in the global dependency ratios rising. The dependency ratio is what economists use to calculate the proportion of the population of the working to non-working percent of the population, meaning that the retirement age people divided by the total working population. And as you can see here in this chart, Japan is the most aged, but actually Western Europe is coming in right behind them. This results in dramatic increases in healthcare expenditures, as we're all familiar with. So I'll ask you a question. It is, what is the main risk factor for lung cancer, do you think? Most people would probably say tobacco or smoking cigarettes, but actually that's not exactly true. Technically, it's aging. You don't see a lot of 30-year-olds, even if they're smoking multiple packs a day, actually showing up with cancer or COPD. It takes time. You need the mutations to accumulate in the DNA, or you need the immune system to become weak enough to allow the cancer to arise. So it's actually aging that is the main risk factor that allows for the damage that was done by smoking to emerge. So as you can see in this chart, disease mortality rates with age rise exponentially, such that even if we, the biomedical science community, were able to cure entirely one of the major killers outright, such as cancer, that would only on a population level increase average healthy lifespan by 2 or 3 years. So if we really want to make a dent or move the needle in public health, we need to find a way to extend healthy lifespan. The current approach of regulators and biomedical research funding entities is the whack-a-mole approach, or the Hydra problem, meaning they're going after one age-related disease at a time, when in reality we should be figuring out what's going on at a molecular and biochemical level that is driving all of these diseases. So here you can see the objective is to not necessarily increase lifespan overall, but to increase the period of health span, the period in which we're in good health. So you can see from this chart that the number of able-bodied people over age 65 in the US determined by whether we're able to delay particular diseases versus whether we're able to slow down aging itself. And if you zoom in, you can see that there are 87 million people still able-bodied over the age of 65 who are able to work and contribute to society and who are not, importantly, drawing upon the medical care system. Whereas only 76 million if we delay cancer, roughly the same with heart disease, and that's only 1 million or so above the baseline scenario. So the way to move the needle is to slow down the aging process itself. And some economists calculated that this would be equivalent to $4 trillion saved over the period, just in the United States alone, which is equivalent to the value of 43% of the gold ever mined. So moving into the second part here, I'm going to elaborate on the process of extending health span and what that really means. So you can see that the objective of medicine for the last century or more has been therapeutic interventions targeted after particular conditions, atherosclerosis, neurodegeneration, cancer, etc. Whereas the new thinking is that we can have a therapeutic intervention that slows down something fundamental about the aging process and thereby attenuate all age-related diseases in one fell swoop. So in the business, we call this squaring the mortality curve, or simply adding life to your years rather than just years to your life. And this is the stated objective of the National Institute on Aging in the United States with a $3 billion budget plus. So in this chart here, you can see on the y-axis is population survival. On the x-axis is years, or lifespan. And there is a threshold horizontally beneath which, if you go below that, you're no longer in good health. So you can see the base case scenario here. Average lifespan in the West is somewhere between 70 and 80 years, and that continues to increase. What we can do with modern medicine is we can engage in disease management. So we're very good at this, and it's very expensive and unsustainable. But basically, we push out lifespan a bit, but it's already in that period where you're in ill health. So you're not necessarily benefiting from quality-adjusted life year gains. With the perfect, most ascetic and disciplined diet and lifestyle, we can extend the healthy period of life, but even then, ultimately, we will age and drop below that good health threshold. If you combine good diet and lifestyle with the advances that I'm going to tell you about in a moment, brought to us by the field of geroscience, we can dramatically extend that period of healthy life, minimize the area under that curve, and ward off age-related disease. And then someday in the future, we can talk about dramatically extending maximum lifespan rather than just median lifespan or squaring the curve, as we say. So in this paradigm, actually through advanced therapies like cell therapy, genetic engineering of the germline, there's really no telling how far we can go. And we've been successful. So as you can see here, we have been, since the 1850s, squaring the mortality curve. We're spending less of our aggregate lifespan in ill health. And so hopefully we can all live our lives like these happy people in the stock photograph are. But of course, your results may vary. So we know that it's possible through the diet and lifestyle approach to extend healthy lifespan. And this is proven to us by the blue zones. So blue zones are areas where there's a high concentration of what are called supercentenarians. These are people living well beyond 100 years of age in relatively good health. And these blue zones are spread all over the world. One in Costa Rica, one in Sardinia off Italy, one in Greece, one in Okinawa in Japan. And many of these are actually islands, you'll notice, because they're isolated from the bad diet and lifestyle of modernity. But we also know that it's not a genetic phenomenon. It's not an isolated population issue, because if you were to take somebody from Okinawa and put them on a Western diet, for example, or move them into the West, they actually age at the same rate as the rest of us. In addition to that, there are genetically determined supercentenarians. So there are genetically isolated populations, such as one in New York, that despite bad diet and lifestyle, continue to survive and outlive their peers and even outlive the doctors who tell them, you know, you should really stop smoking cigarettes and drinking whiskey every day. And these people are really, they've won the genetic lottery. So we've made efforts to identify the polymorphisms or genetic differences in these people that allow them to resist age-related diseases. And drugs are being developed to harness that information. So what has made human beings and medicine has quested after immortality or life extension since the beginning. You go back to the Epic of Gilgamesh. It was about this Sumerian king who went traveling all over the earth to find a substance, a plant that would grant him immortal life. So we've been interested in this for millennia, but it's left the realm of science fiction into the realm of reality thanks to the revolution of molecular biology and biochemistry in the last half century or so. We've been able to pinpoint the fundamental molecular mechanisms that are driving the aging process. And we've actually been able to partially reverse this process in mice. So I won't get into the details of the science today, but suffice to say that there are orthogonal, multiple interventions that we can combine with synergistic effects in various model organisms in the lab. And in the next couple of years, we will see the first drugs targeting these fundamental pathways of aging entering into the clinic. So only in the last 5 or 10 years has aging and geroscience become sort of an interesting buzzword, and people are excited about it. It was previously considered intractable, or aging was just a process of entropy, one that we could not necessarily intervene in. But with some proofs of principle lately, it has gotten some pretty high-profile groups into the field. So for example, the National Institute on Health, which has a $34 billion research budget, it's the largest in the world, has created a trans-NIH geroscience group, and they're having a conference every year to discuss this. Human Longevity Inc. is a company based on genome sequencing by Craig Venter, who led the private sector effort to decode the human genome in the early 2000s, culminating in 2001. Unity Biotechnology in the Bay Area is developing drugs that eliminate a certain type of zombie cell called senescent cells. I'll tell you more about that in a moment. Novartis, one of the largest and more innovative pharmaceutical companies, has recently spun out a company called Restor Bio that is working on an aging mechanism. There are a number of venture capital funds and company builders in this space. So the Longevity Fund, Apollo Ventures, where I work, Juvenescence. These funds are all focused on longevity and the fundamental biology of aging. And in Apollo, we've built our fair share of companies going after each of these various hallmarks of aging. Jumping into the fray has also been Google, the tech company, with a company called Calico, or California Life Company, that they've backed with $2.5 billion and a partnership with AbbVie, the pharmaceutical company. And they've recruited a lot of high-profile aging scientists into this company, but they're very secretive. Not many know exactly what they're up to. And there are prominent labs all over the world now getting very excited about the prospects of slowing down the aging process. So now I'm going to sort of paint a picture and make this real for you with some case studies. What exactly has been done that excites the field to demonstrate the proof of principle that we can actually extend healthy lifespan in mice and probably humans? So the first intervention is actually quite bizarre. It's called Heterochronic Parabiosis, which basically means you take an old mouse and a young mouse, and you surgically link their circulatory systems such that they share blood. And what results is the old mouse gets younger and the young mouse gets older. And you might have seen Silicon Valley, where they have blood boys following you around donating blood. That's not going to be necessary, we hope. The objective or the goal here is really to isolate the proteins circulating or small molecules or whatever it is in the old blood or the young blood and isolate that and turn that into a drug. Another interesting intervention has been senescent cells. So senescent cells, as you can see here on the right, have this fried egg sort of phenotype as compared to these normal, healthy, thin, spindly fibroblasts, in that senescent cells are aged and their telomeres are damaged due to replicative senescence, or they have some other type of cell stressor, and they effectively become zombified. And they can cause other nearby cells in a paracrine fashion to become senescent as well. And what that results in is a chronic pro-inflammatory cytokine load called the SASP, a secretome that accelerates aging and the risk of cancer throughout the entire body. And what has been demonstrated by a number of groups that genetically or pharmacologically, one can ablate these senescent cells and extend lifespan, healthy lifespan, in mice by up to 30% here. In addition, they can rejuvenate aged mice. So for example, you can see on the left, there is an aged mouse with a hunched back or kyphosis, cataracts in the eyes, arthritis of the joints, thinning fur, et cetera, many of the hallmarks of an aged phenotype. Whereas if you administer some of these senolytic compounds, you restore eyesight, no hunching, no arthritis, and the fur is healthy and glossy again. So there are a number of companies working on this, one of which is Unity Biotech, which is going into clinical trials now for osteoarthritis, reading out in the first quarter. Another is called Clero, which is an Apollo Ventures company with a peptide that achieves similar results, although I'm biased. I think they're a little bit more advanced. Another interesting approach that you've probably heard about, most people who know about aging have heard about telomeres or telomerase, the enzyme that extends telomeres. Well, it was demonstrated by Maria Blasco's lab several years ago that using a viral vector, an adenoviral vector, AAV, you can deliver telomerase, extra copies of the gene telomerase, into the mouse, an aged mouse, and it will rejuvenate the mouse, and they will live about 20% longer. And the animals look younger in pretty much every way that you look. Many people were concerned that this approach of reactivating telomerase would increase the risk of cancer. This is true with germline engineering, but it's not apparently true with AAV, viral vector-mediated gene therapy, because the construct is diluted out should the cell start to proliferate. Another approach is one of my personal favorites, known as caloric restriction. So essentially, it's free. You can do it today. And it's been known for thousands of years, since Hippocrates, or certainly most religions advocate fasting from time to time. It's basically you reduce your caloric intake by a third, and animals such as mice live a third longer. Although most of us don't have the fortitude to actually force themselves to cut caloric intake by a third every single day. So another approach is intermittent fasting, which is what I do. You can sort of skip a meal or eat within a narrow period of the day. But really what we want, what we're all looking for, is to have your cake and eat it too, so to speak, meaning you could eat whatever you want and still not get fat or metabolic syndrome, and you would retain the metabolic profile of a young person. And several years ago was one of the first attempts at this approach by a company, somewhat controversial, spun out of Harvard, called Sertris, by David Sinclair, that was acquired by GlaxoSmithKline for $720 million all in biobucks. And that didn't work out, as a lot of biotech companies don't. But it definitely illustrated that there's a lot of commercial interest in this field. And another molecule more recently studied, called NAD, or NR, as a precursor, that also is a naturally occurring molecule that mimics caloric restriction or fasting and extends lifespan across model organisms. So let me ask you a question. Seems obvious. Which monkey depicted here is the older one? Probably the one on the right, right? Well, actually, it's a trick question, because they're the same exact age. It's just that the monkey on the left underwent caloric restriction for most of its life. The monkey on the left wasn't quite so happy, maybe slightly more aggravated, but he was definitely more active and so on. It's really amazing. If you look at videos of these monkeys, they're the same age, but the one on the left is so clearly younger. So you've probably heard of resveratrol, which is one of these caloric restriction mimetic molecules. It actually occurs in grapes and blueberries, and some believe that resveratrol is the reason why a little bit of red wine is salutary or lifespan extending. That's probably not true. That seems to be propaganda from the wine industry, because you have to drink so much wine to get a physiologically relevant dose of resveratrol that it's just not feasible. But I know some of you will take that as a challenge rather than a warning, but that is your prerogative. So another of these really interesting geroprotective molecules, or lifespan extending molecules, is called rapamycin. So rapamycin was discovered on Easter Island, the island of Rapa Nui, where it derives its name, and it was almost lost again. The chemist who identified it, basically his research program at a pharmaceutical company was shut down, but he knew there was something special about that molecule, so he hid it. He stashed it in the back of his freezer for years, and it was rediscovered. Fortunately, because rapamycin is one of the most important drugs for immune transplantation. When you get a donated kidney or liver or organ, oftentimes the immune system will reject it in graft versus host disease. But if you administer an immunosuppressant such as rapamycin, that probability is lowered. But it more recently was demonstrated that actually rapamycin, even when administered late in life to aged mice, extends their lifespan by about 13% median lifespan extension among genetically heterogeneous mice. And this was a really interesting finding, and it has spurred a lot of research in this field going after the protein target, the complex, mTOR complex 1 and 2 of rapamycin. And the objective of this research is to develop analogs or structural derivatives of rapamycin that do not come with the side effects such as immune suppression or metabolic syndrome and dyslipidemia. And Apollo Ventures actually has a portfolio company known as Aeonian Pharmaceuticals that has made great strides in achieving that goal. What's another interesting drug, an anti-aging drug, has actually been on the market for many years, and it's known as metformin. Metformin is the first line of defense that doctors will prescribe to diabetics or people with prediabetes because it lowers blood sugar. But metformin has also been demonstrated to extend healthy lifespan in mice quite dramatically. And so a gentleman by the name of Nir Barzilai at Albert Einstein in New York has received permission from the FDA, the Food and Drug Administration, to initiate a clinical trial in aged adults to determine whether metformin can actually slow down the aging process, or the latency from the first morbidity to the second morbidity. Basically, when you have an age-related disease or any disease, you're more likely to get another condition shortly after. And if this drug can extend that latency period, that is used as a proxy for extending healthy lifespan. So a lot of these caloric restriction mimetics and the last several compounds I've mentioned have a common denominator, which is a cell biological process called autophagy, or the Greek for self-eating, which is whenever a cell is under a bit of stress or fasting, there is a lack of availability of nutrients, or under exercise conditions, the cell will break down its internal components. It'll consume them and then rebuild them. So organelles such as the mitochondria or the endoplasmic reticulum will be broken down and rebuilt in this process. So it's been demonstrated in various model organisms that you will not achieve the lifespan benefits of fasting or exercise if you have genetically ablated or deleted or inhibited the process of autophagy. So autophagy is required genetically. It is necessary for the lifespan extension for many of the best characterized geroprotective interventions. So that led Apollo Ventures and a couple others very recently, just this year, to skip the middleman and look for new drugs that will directly activate autophagy in various subtypes of autophagy. We have very interesting results so far, so stay tuned. So coming into the last part of my talk, I'm going to conclude with what is called the holy grail of geroscience, which is understanding and harnessing germline immortality. So immortality, it's actually a solved problem, at least for cells. A gentleman by the name of August Weismann, one of the greatest developmental biologists, contributed to a theory known as the disposable soma theory of aging, which is basically you have the soma, which is almost all of the cell types in the body, except for the germline, the reproductive cells. And the idea is that the soma is basically just a glorified meat suit that carries around and does the bidding of the germline, which are the reproductive cells, the sperm and egg and the stem cells that make them up. So it's been demonstrated that the primordial germline cells, the gonads that you all possess, were actually originally inherited from the zygote, which was created from the fusion of sperm and egg of your parents. And the gonads in their bodies were created from their parents, and so on and so on. So the gonad cells, the germline, the reproductive cells, they're actually technically immortal. They go on all the way back to the dawn of life on this planet 3.5 billion years ago. So not only has cellular aging been solved, it's actually been solved for billions of years now. So it's not a matter of physics or entropy that we cannot slow down aging. It's just a matter that biology has structured the soma, in humans at least, not to be immortal, or not to be able to tap into this incredible regenerative capacity that exists in the germline. It's also been shown several years ago by Shinya Yamanaka and others, a Nobel Prize was awarded for the process of induced pluripotency, which basically means you can take any cell type, such as a skin cell from a person, treat it with a bit of genetic tomfoolery, and you can rejuvenate the cell, bringing it back to an embryonic stem cell-like state. And that eliminates or deletes or clears away all of the markers of cellular aging. And then you can take that cell and transform it into basically any cell type in the body that you want. So that allows you to go from a skin cell to an embryonic-like stem cell that is totipotent or pluripotent, and therefore into any cell type that you want. So a liver, neurons, etc. And this process meant the breaking of the Weissman barrier, the barrier that August Weissman described, that describes that the soma cannot actually return to pluripotency as the immortal germline is. So that's on a cellular level, that's great, but what about whole organisms? Well, there are actually organisms in existence, multicellular organisms, that do not really age with the passage of time. One of them you can see here with the adorable googly eyes is the regenerating planaria worm, where you can slice off its head and it'll just grow right back. Another is the immortal hydra, this sort of sea anemone-like creature that is not more likely to die as time passes on. It's functionally immortal. Interestingly, not a lot of people realize this, but babies, human babies in the womb, and even shortly after birth, have incredible regenerative capacity. So for example, if a baby has a heart condition and it's noted with ultrasound, a physician can actually go in and perform in the uterus surgery on the tiny heart of the baby and sew it up, and it'll heal in a scarless way as though there was never any surgery at all. In addition, when a baby is born for the first several days or perhaps weeks, a little bit of the finger, if it's lost, if it's sliced off, can actually regrow without any scar. So we actually latently possess these incredible regenerative capacities, but for some reason, evolution has decided to shut them off as we develop. And probably one of the theories is that, well, we're always kind of on the knife edge of a stem cell turning into a cancer cell because they're both semi-immortal types of cells, and you don't want too much regenerative capacity late in life after a lot of damage has been done because you risk creating cancer. So aging, the takeaway is that aging is not a law of physics. It's not entropy. It's a solved problem in cells and multicellular organisms. We just need to understand how it works. So in the last decade or so, there have been over 40 interventions described that can extend healthy lifespan in mice, for example. Some of them are genetic, so turning up or down the volume of a particular gene, such as growth factor signaling. Others are pharmacological, so certain substances such as senolytics or even a molecule in green tea called EGCG can extend lifespan a little bit. So as you can see from this list, most of these interventions so far that extend healthy lifespan, they're genetic interventions. These are not available to us as developed adult humans, at least not until gene therapy is working. So we really need to focus on the pharmacological therapeutic approach that we have today. And that's one of the objectives of Apollo Ventures and groups like it, where we build biotech companies spinning out technology from universities that target the fundamental molecular mechanisms of the aging process. So a couple of them are listed here. One is Aeonian Pharmaceuticals, which I mentioned, which has these best-in-class rapamycin analogs. Another is a company called Clera in the Netherlands that has a senolytic, or a molecule that ablates senescent cells and can extend healthy lifespan in mice and ameliorate various models of disease. Another is Samsara Therapeutics, named for the Buddhist cycle of reincarnation, because this company is interested in proteostasis and the mechanisms by which cells are able to turn over their internal components and renew themselves. Another is Telos Biotechnology, that is leveraging the power of telomere biology to rejuvenate cells for cell therapy, such as CAR-T, to enable indefinite expansion of the population of these cells. So in cell therapy, one of the limits in delivering these cells to patients is actually you just can't grow enough of those cells fast enough to deliver the therapy to a patient who needs it urgently, such as a cancer patient. And we have a method to rejuvenate these cells outside of the body and then reimplant them. And many other companies in stealth mode. So I'll close here with my personal hopes for the future of geroscience. The question is, why am I doing this? What do I hope to achieve? So first of all, to live as long as possible in good health and to be apprised of those advances and not spend the last few decades of life in poor health, which is actually the base case today. Not a lot of people realize that, or they're in denial, but most people spend 5 or 10 years, sometimes more, with multiple chronic diseases at the end of life. And personally, I think it would be a great outcome if I could die at the age of at least 110 in one of 3 possible ways, and bear with me here. Peacefully in my sleep, okay? Possibly in a tragic surfing accident in a beautiful warm locale. Or I would also accept being killed by the jealous ex-husband of a new lover. So with that, I thank you for your attention, and I can take questions.